sort of the next practical is observation of specimens taken from electron microscope. Right. So I have some electron microscope images, right, that I'll show you using the project. Okay. Come to the resource book. Come back to the resource book. Next topic, online people also come back to the resource book. Page number 23, the electron microscope. Electron microscope. The limitation imposed upon the resolution power of the light microscope by the wavelength of light, right? So the limitation is what? The wavelength of visible light is lower, right? So there's a limit to the clarity we can get, right? Magnify there's a limit that we can magnify, but we can't nicely see. Okay. The resolution power is inversely proportional to wavelength. Now, it's been erased, but you know, right? When wavelength increases, the resolution power will decrease. So what is the solution? To find a wave type with a what? Smaller wavelength, right? Smaller wavelength. So due to this, the scientists consider the use of other forms of radiation with comparatively shorter wavelengths. As a result, electron microscopes were developed, and in electron microscopy, a beam of electrons is focused to the specimen or to its surface, right? So this statement is important. We can either send electrons through a specimen, or you can just bounce off, right? Bounce both those things can be used. Okay. Electron microscopes have revealed many organelles and other subcellular structures that were impossible to resolve. That means nicely see, right? Resolution with the light microscopes. Okay. So as I mentioned earlier, we can use the electrons in two ways. Either we can send them through or bounce off, right? So based on those two methods, we have two types of electron microscopes. First one, transmission electron microscope, that uses what principle? We are sending electrons through. Transmission. Okay. It is used to study the internal structure. So if you have a cell, you are going to shoot electrons through it, right? Then you can see the inside. That's the logic. Okay. The beam of electrons is passed through a thin, specially prepared slice of material. A very thin specimen is used, and the specimens are stained with heavy metals which attach more to certain cellular structures than others. I'll show you some images, then you can see how this staining with heavy metals, right? We sometimes have to use gold, right? Because that's why, other than the fact the microscope is very expensive, right? The microscope is very expensive, and the staining is very expensive as well, right? Specimen you need a very microscopic amount of gold, but more than the value of gold, the staining procedure is, let's say, complex, right? So electron microscopy in general is a very expensive procedure. We usually limit it to observe viruses, to study the structure of viruses, right, at this stage, right? Because light microscopy is kind of sufficient to study the bacteria. Right? Okay. Image reflects the pattern of electron passed through the specimen displayed on a screen. While electrons pass through the specimen, more electrons may get displayed in regions where the structures are densely stained, right? So if we are using a heavy metal die, the structures that get, where more gold gets stuck to they will sort of attract more electrons. They will be heavily sort of colored by electrons. Okay, I'll show you. Then that is the transmission electron microscope. The second type is scanning. What are we going to do? We are going to bounce electrons over the surface. So what can we observe with a scanning microscope? The surface features, not the internal structure, the surface features. So as they say, a fine beam of electrons are reflected from the surface of the specimen. Specimens are mostly coated with gold, right? Again, the surface is coated with gold, so the reflection can happen nicely. Okay, any question? Here the specimen scatters many electrons, whereas others are absorbed, right? Scatter venevat, absorb venevat. Okay. This instrument is ideal to observe the surface view in 3D appearances, right? Nicely surface එක 3D view එකක් බලා ගන්න පුළුවන්. What are the like outer plasma membrane and what are the structures attached to the plasma membrane? ඔය වගේ දේවල් using the scanning electron microscope we can observe. Okay. This next chart is very important, right? Very important. Put a star mark or whatever. This comparison will be questioned in MCQs, structured, right? Very common. So we have to compare light and electron microscopes. In light microscopes, what is our focusing instrument? Glass lenses that refract light. Okay, glass lenses. In electron microscopes, what do we use to focus? These are electrons. Can we use glass lenses? No. We are using magnets, right? Magnets that can sort of focus, concentrate the electron beam, magnetic fields. Okay. So magnetic lenses, we consider them as lenses that are made of magnets. Okay. Then image is directly detected by naked eye. We can place our eye and we can observe, right? But now there are light microscopes that can be connected to a computer, right? So those in those pathology labs, as I told you, there are microscopes that you can plug in to see on a screen, but they are still light, right? They are detected by a sensor here. Still light microscopes. If you want to, you can put your eyes also, right? ඉතින් බෑනේ දවසම මෙහෙම කරගෙන ඉන්න ගියාම consultant pathologist කෙනෙක් මෙහෙම මොකක් කරේ මෙතන this eye will get a spasm or something, right? So they are using a screen, but still light is used. But there are no options in an electron microscope. You definitely have to look at everything through a screen, right? Because we can't see electrons, right? Living and non-living objects can be observed by what? Light microscopy. So this onion peel has living cells still because they are still in water. No? So for maybe about a few more hours until they get dehydrated, they are still living. Yes. Okay. But in electron microscopy, because of two things. මුලින්ම මැරෙනවා when they are coated with those heavy metals, right? Anyway, they are going to die when they are coated with those heavy metals. Secondly, when the electrons are going through them, their cellular structures are going to get damaged, and anyway, those cells are going to die. So nothing can survive electron microscopy. So you can only observe what? Dead specimens, right? The cells have died. Okay. So what do you mean by non-living here? Like we are not taking dead parts, but because when we are preparing it to be observed, it will be killed, right? Actual color of the image can be observed. What do you mean by color? It's like visible spectrum එකේ තියෙන different wavelengths, no? Blue, red වගේ තියෙන visible from I think red to violet, the wavelength changes, right? එකක් නෙති. So we can observe that. But electrons that will be detected by a sensor in the microscope, and we can give artificial colors. There are softwares for electron microscopy. We can ask them to give colors to certain structures, right? So artificial colors can be put so that we can nicely identify, or for teaching purposes, we can put colors using software. So actual color can never be observed in electron microscope. Dyes are used to stain, right? So when we are in the next part, that is the cellular basis, I'll tell you about this staining, right? There are several dyes that we can use to color these cells nicely so we can easily observe them, right? Usually we are using this onion and raw here because why? They have a nice natural color, right? Pink, raw here has also like a purplish color we can see. But if you observe some, let's say, bacterial cells, they look colorless, right? And just water droplet, they look colorless. We have to use some special dyes so that we can color them in the special structures in different colors so that we can observe and identify the structures. Okay, those are called stains. The stains are of two types, as the dye or pigment stains or heavy metal stains used in electron microscope, right? So you can just say stains are of two types, as pigment, that means just color, no, that we can see, pigment stains, and yes, heavy metal. If you want, you can write down. Pigment stains are there, heavy metal stains are there. Okay. Any questions? Yes, you have questions in the tube. Yes, let's do those quickly. 5 more minutes, no? Let's do those as the at the beginning of the next class. Right? එතකොට ඒක warm up paper එක විදිහට use කරන්න පුළුවන්. Right? Let's just go through the electron micrographs. Okay? There's one essay that we haven't marked, no? Water essay we haven't yet marked. Right? I'll give a marking scheme for that. This week I'm giving another essay. Did I give any essays last week? No? Okay. It's clear enough, right? That is again type of focusing. Okay. Right. Right. Electron microscopy. Let's start from the beginning. There was nice. So anyway, what we see in this black and white are electron microscopes. I'll show you. Hold on. Ah, yes. Okay. This is. Can you see this diagram? It's like surface of the moon, right? This is not a bhima. It's not a cavity, but it's a sort of a, let's say, like a elevated round shape, right? That is inside your cells. What do you have? There's a nucleus, right? That surrounding the nucleus, we have a separate sort of a covering for that nucleus that is called the nuclear envelope. Next week we are starting. Okay. So what are we seeing? This is a surface view of that, no, nuclear envelope. So if you are seeing a surface view, what kind of a, you can see, right? What kind of a microscope are we using? Electron. What kind of electron? Scanning, no? Because why? We are seeing the surface features. They are, that means the electrons are being reflected, right? So what does it say? Yes, scanning electron microscope, 34,000 magnification, right? 34,000 magnification. This is a transmission electron microscope. Can you see? This is that same envelope or the membrane. Okay, online students, you can see, yes. This is that same envelope, but observed by what? What microscope now? This is transmission. Why? Can you see? It is like a sliced view, no? Imagine that membrane has been sliced. So this is that new this roof-like structure, but now we have sliced it this way, so you can see the two layers. Can you see the two layers? Because why? The electrons are going through it. Transmission electron microscope. Okay, so you can see other structures inside the cell as well as this, as if the cell has been sliced. Yeah. Usually, transmission electron microscope has higher magnification. Okay, this is about 59,000 magnification. Okay, transmission electron has somewhat of a larger magnification. These are again transmission electron microscope images. You know what the rough endoplasmic reticulum is? No? Yeah. So it's a massive network of membranes inside the cells. This is the nucleus, okay? That is where all the DNA is. This is the membrane network surrounding it. Have you heard of ribosomes? Ribosomes කියන දන්නවද? Ribosomes are මේ dot 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 ගොඩාක් තියෙන, right? Those are our protein making factories, right? So the rough endoplasmic reticulum has ribosomes stuck to the membrane. Can you see dots? Those are ribosomes. Okay. Let's see if there's another scanning. We usually use transmission to study structures because why? We can see a nice cross-sectional view. These are all transmission, okay? This is a phagocytosis. This is a cell eating another cell, this process, right? Let me just check what are these. What is B is a bacteria, lysosome. Ah, yes. So this, what is happening? This cell is eating other cells. So this is a big cell. I think, what is it? It's not given, right? This is a bacterial cell, small one. Can you see? This is, imagine this is one of the white blood cells in your body, right? So this is a bacterial cell that is going to be engulfed. These are other things that have been other bacterial cells that have been previously eaten. Right? Okay. So this is again electron microscope, right? Transmission electron microscope. Okay. This is a practical, okay? The practical guide, you have a practical called observation of electron microscope images. In your school labs, you will have this laminated, some electron microscope images would be there, but right? Those are standardized ones. You can clearly see the structures. Okay. Lab එකේ ඇති laminate කරපු file එකක් electron microscopes. If you go to school, okay. Some structures that you know mitochondria, yes. These are again mitochondria observed from transmission electron microscope about 34,000 magnification. Again another mitochondria. Okay. This is a light microscope, right? Medium power. B. What are B? B are blood cells. These are muscle, I think. Yes. You can see some glycogen, right? Glycogen, stored in muscle, right? This is what? This is a light electron microscope under medium power, right? This is a light electron microscope image. Okay. Screenshot tag, right? You can take digital from the screen. You can take photos, right? Some things very rare scanning electron microscope images. These are all high power images of cells dividing, right? So this is before a cell is dividing. So we'll talk about cell cycle. At the end, what do you have? We have two cells. Nice, right? We have nice two cells. This is, you can see these under light microscope, high power, maybe 1000 magnification. Okay. I'll send to the group some more better scanning electron microscope images if I find. Okay. For now, I think this is enough. Yes. So if you like, you can stay and do the fructose practical or anything else. You can try glucose or whatever. Any questions? Online people? Any questions from what we did? Any feedback from you? Did we miss anything that you thought would be done? Yes. I think the interactive part was a bit missing, but it's your fault. Okay. Yeah. So anyone? Oh yeah. So finally, what did we do today? Matakada, matakada. What was the first experiment that we did? Online people also pay attention. First, we tested. Kya? No, glucose, right? Which is a reducing sugar. Now you can finally see it has precipitated, right? Which is this is what Cu2O, cupric, cupric, right? Oh, plus whatever Cu2O. I'm not a chemistry teacher. Okay, Cu2O. This is the precipitate, right? That has nicely settled, right? So can you see? So the solution color decreases when the precipitate forms. You can online people, can you see? Hold on. Brick red precipitate underneath. Alannato, you can pass. Right. Second one, what did we do? Reducing sugar. So non-reducing sugar, sucrose. What was the process? We add an acid, we heated, then we neutralized it. Then we did the Benedict test. Can you see? Online people, can you see? The brick red precipitate underneath. Right. Next, what did we do? Starch or biurate? Biurate. Egg white, what was the color supposed to be? With time, this solution gets reacts with oxygen, and because it has copper, it changes into some weird colors. What was the proper color that we observed? Purple color we observed. Make a denna bitter halagat. Right? Okay. If you want, you can. Right. Then what was next? We observed starch. Right? Iodine test for starch. Originally, brown color iodine turn black or dark blue starch. Right? Then we did Sudan 3. Can you see the water layers nicely separated? Water layer and the coconut oil layer, which is a lipid, stained with the non-polar dye, which is Sudan 3. Okay. Right. And also this another sort of solid state of starch, right? To wheat flour and also to a slice of bread. We did the iodine test, got the same color change. These, I mean, from practicals, there's likely to be an MCQ, okay, in the A-level paper, and a small part of a structured essay, but in your first and second term tests, there's a high chance you might actually get a big part of a structured essay and maybe 2-3 MCQs. Okay, so that's why I took this sort of the effort to do this. If you like, you can, yeah, if you like, you can come and do the fructose thing, or then if not, you can go home.